This will be a walkthrough of the AQA GCSE Combined Science Trilogy 2022 Biology Higher Tier Paper 1. Question 1. Bacteria can cause a variety of diseases in humans. What are two similarities between a bacterial cell and an animal cell? Tick 2 boxes. So the information that's given to us in the question is talking to us about two types of cells, a bacterial cell and an animal cell, which we have knowledge of. These are two different types of cells. So a bacterial cell is an example of a prokaryotic cell, and our animal cell is an example of a eukaryotic cell. So what we want to do is look at each of these structures that we find in these cells and see which both relate to bacterial and animal. So the first one, both have a cell membrane. That is true, they both do. So that should definitely be an answer. Both have a cell wall? No. Our animal cells don't have a cell wall, so that can't be right. Both have a nucleus? No, our bacterial cell doesn't have a nucleus, so that can't be correct either. And both have a cytoplasm? Yes, they both do, so that one is also correct. Question 1.2. Salmonella food poisoning is caused by bacteria in food. Give one symptom of salmonella food poisoning. Do not refer to vomiting or diarrhea in your answer. 1 mark. So we've got to think about what else can happen if someone is suffering from salmonella food poisoning. Well, one common symptom for quite a variety of diseases is going to be a fever. So fever should be an answer because that's one of the responses to try and get rid of whatever pathogen is in your body. What we could say for another answer is that you might have abdominal or stomach pains. Question 1.3. What is the name of the first antibiotic developed? 1 mark. Well, thinking about bacteria again, because antibiotics are there to kill bacteria, and the first antibiotic that was developed was by Alexander Fleming, and the name of it was penicillin. There are no other antibiotics that need to be known for the specification other than penicillin. A child with a severe bacterial infection was given a course of antibiotics. Figure 1 shows how the concentration of live bacteria in the child's body changed when taking the course of antibiotics. So looking at this graph now, we've got x-axis time in days, got the y-axis concentration of live bacteria in the body in arbitrary units, and it can tell us where the course of antibiotics started. 1.4. The concentration of live bacteria in the body continued to increase after starting the course of antibiotics. Suggest one reason why. So going back to the graph, we can see at day 0 when the course of antibiotics started, the concentration increased slightly and then it began to decrease almost up to 1 day and then continued decreasing up until the end. So you've got to think about why there was this increase. Well, one of the reasons may be depending on where the antibiotic had been given. Usually it's given and ingested, and then the place where the live bacteria were. So maybe the antibiotic hadn't reached the part of the body where the bacteria was. Usually it might take some time for the antibiotic to reach there, so maybe there was a bit of a delay between those 2 points, where the antibiotic was given and where the antibiotic needs to work. The other thing that could be said is the bacteria, even though the antibiotic was given, the bacteria just continued reproducing after the course of the antibiotics had been taken. Question 1.5. After 3 days of taking the antibiotic, the child felt better. There was still bacteria in the child's body. Why did the child feel better Tick one box. Bacteria had become immune to the antibiotic. No, that wouldn't be the case because the child wouldn't feel better. The child had become resistant to the bacteria. Again, same issue. The child would not feel better yet. And there are fewer toxins in the body than at day 0. That does make sense. The child still feels better, but I said fewer toxins because remember that bacteria are supposed to be producing toxins. 1.6. Suggest why doctors do not give antibiotics to patients with minor infections. Well, what's going to help us with this question is some of the possible answers from the previous question. Because one thing that bacteria are able to do are to become resistant to an antibiotic. And that is one of the reasons why there is a strict prescription that is given when anyone is on some sort of medication. So it is to reduce the chances of developing these resistant bacteria to any antibiotic. A reminder, the reason why bacteria become resistant is through mutation of their DNA. Figure 2 shows blood viewed using a microscope. So we've got a figure here. It's got different parts of blood. What we can do using our knowledge is label them. So A, big circles with a dent in there. That looks like our red blood cell. B, larger circles. And they look like they've got some stain in the middle. Would be our white blood cells. C, it looks like the liquid or fluid part of the blood. And that's pointing to the plasma. And D has got to be the small little fragments, which are the platelets. 1.7. A vaccine will stimulate the production of antibodies. Which part of the blood in figure 2 produces antibodies? Tick one box. Well, it's got to be what part of our blood is supposed to be fighting infection. That should be the white blood cell, so it should be B. Which part of the blood in figure 2 starts the clotting process? Or the part of the blood that starts the clotting process is those small fragments, which are the platelets, so it should be D. Question 2. This question is about cell division. 2.1. Write the biological structures from the box in the correct order of size. So we need to go from smallest to largest using these terms. So if we draw a cell first, what we can find inside of our cell is a nucleus. And inside of that nucleus, we find chromosomes. And chromosomes have small sections of them called genes. So looking at our diagram now, we can figure out that the smallest should be gene, then after that should be chromosome, after that it would be nucleus, and then the largest will be a cell. Figure 3 shows how a fertilized egg cell can produce specialized cells. Okay, our fertilized egg cell produces embryo, and then it goes through process A to give us red blood cells, muscle cells, and nerve cells. 2.2. Name process A. So it's these embryos going to form these different specialized cells. Well, we can't call it specialization. There is a specific term for this process. It's called differentiation, the process in which a cell becomes specialized. 2.3. How many cell divisions are needed to form a 16-cell embryo from the original fertilized egg cell? So it seems like we need to do some calculation because we're looking for a number here. So let's remind ourselves, a fertilized egg cell to develop into an embryo must go through the process of mitosis, which is a form of cell division. So if we're starting off with one cell, at the end of mitosis, it should form 2 cells. And that technically would be one cell division. If that happened again, we would then end up with 4 cells. So after 2 cell divisions, it would be 4 cells. Again, one more cell division, and then that should mean we have 8 cells. Last time, we should then end up with 16. So now we've gotten our 16 cells in our embryo. We just need to count the number of cell divisions that we get from there, and it should be 4 cell divisions. Now, there is a formula that we can use to actually help us with this, and the formula is 2 to the power of n. So we know then that our answer, if we do 2 to the power of n, should be 16. So we just need to know what that n will be, where n is always the number of cell divisions. So 2 to the power of 4 would give us 16. 2.4. In the humans, a fertilized egg cell contains 23 pairs of chromosomes. How many chromosomes will there be in each of the embryo cells? Well, our embryo should have a full set of chromosomes, half from mother, half from father, because it just came from a fertilized egg cell. So, if it tells us that a fertilized egg cell contains 23 pairs of chromosomes, we need to be careful and remind ourselves what is 23 pairs. That's the same as 23 times 2, which is 46. So the same number of chromosomes in the fertilized egg cell should be in the embryo. So it should be 46. 2.5. Figure 4 represents a cell cycle for a human embryonic cell. Describe one change in the cell that occurs during each of the stages of the cell cycle. Okay, so it's told us that stage 2 is mitosis, but we don't know what stage 1 and stage 3 are. So using our knowledge now, what we can do is we can annotate what happens in stage 1. Well, it's the biggest stage, so it's where the growth of the cell is going to occur. Stage 3 is just after mitosis, and that should be cytokinesis. But let's expand on what happens during these stages. So when we mean growth, 2 things really actually happen. The DNA replicates, and the structures that we find inside of the cell replicate. So there's an increase in the number of our organelles that we find inside of our cells. So for example, the number of ribosomes, the number of mitochondria will increase in the number. We know what happens in mitosis. That is where our chromosomes are involved. And realistically, the chromosomes should be moved to opposite ends of the cell, preparing for the cell to actually divide. In stage 3 in cytokinesis is where 2 things happen. The cytoplasm and the cell membrane divide. So, we've answered our question, we just need to put it in the correct space now. So we need to just put one change for stage 1. So we can just say increase in the number of organelles. In stage 2, you can't just write chromosomes. We have to say what happens to these chromosomes. The chromosomes are pulled to opposite parts of the cell. And in stage 3, you can just say that the cytoplasm divides. Cell division is important in the growth of multicellular organisms. 2.6. Figure 5 shows the mean height of boys and of girls from birth to age 18 years. Okay, so we've got a graph that we need to analyze, most likely here. Compare the growth of boys with the growth of girls. Use data from figure 5 in your answer. 6 marks. So we've got to really go through this graph carefully, try and see how much we can get from it. Remember, the command word is compare, so we need to think about the similarities and the differences between what we see from the graph. And we have to use values from the graph also. So looking at the graph, you can see that for both boys and girls, there is an increase from 0 years up until 18 years. However, there are some slight changes that occur. So right from the start, 0 years, you can see that boys' height is slightly higher than girls, so they're taller than girls from 0 years. But we can see that that trend continues like that all the way up until somewhere between 10 and 11. If we look carefully, we can actually figure out that that is age 11. So that's already one thing that we spotted. There's an increase in this height where boys are taller than girls from 0 years to 11 years. What we can see that happens at 11 years though is that the lines cross over each other. So that means that at 11 years they're actually the same height. And we can state that height if we need to, 140 centimeters. But after 11 years, we can see that the height of girls is greater than the height of boys. So we can say that after 11 years, girls are taller than boys. But where does that go up to? We can see that that goes up to age 14. So between 11 and 14, girls are taller than boys. But what happens after 14 years? Well, at 14 years, you can see that the lines cross over again. So that means that they're the same height also at 14 years. And again, we can say that mean height is 160 at that point. But after 14 years, what happens? Girls stop increasing. The line goes horizontal, so their height doesn't increase anymore, or doesn't decrease at all. Whereas for boys, from 14 to 18, their height is increasing still. So let's remind ourselves, what have we gotten from this graph now? We've analyzed and spoken about the boys' height being higher, greater than girls from 0 to 11 years. We've spoken about the fact that the heights are the same at 11 years. We've spoken about the height being the same at 14 years. We've spoken about girls being taller than boys between 11 and 14. And we've spoken about, at the end, there is an increase in boys from 14 to 18, but no increase from girls from 14 to 18. Lastly, what we could do is we could think about the differences in the height and where that difference is the greatest. We can see the lines are further apart at age 18. And what we could do is a bit of calculation there if we don't have enough information. So let's formulate our answer now. From 0 to 11 years, girls shorter in height than boys. Boys and girls at the same height at ages 11 and 14. Between 11 and 14, girls taller than boys. Between 14 and 18, boys taller than girls. Girls stop increasing at age 14, while boys stop increasing at age 18. Okay, and what we can do is we can throw in this difference in height, where it's greatest. So largest difference in height at age 18 years. And what we can actually do, as I said before, is we can find out what that actual difference is. So if we read off to our x-axis and y-axis, we can see that the height for boys at age 18 is 180, and we can see that the height for girls at age 18 is 162. So the difference is actually 18 centimeters. So that will be the largest difference in height between boys and girls from this graph. And there is our answer, and it's enough to get us our full marks for this question. If we want, we can also talk about the rate of their growth, so how fast do they grow. And for us to figure that out, we would need to look at how steep the lines are. So let's look at a part where there is a big difference in this rate of growth. And we can see between ages 11 and 14, there is a clear difference there. So because the graph for girls is steeper than for boys, what that means is that the girls are growing faster at that point compared to boys. So we can say then that girls grow faster between ages 11 and 14 compared to boys. 2.7. Give one way that cell division by mitosis is important in fully grown animals. Right, so they put in fully grown for a very important reason. So reminding ourselves of why mitosis is important overall, we know from before that it's a form of cell division, but it has three roles. One is for growth, one is for repair, and the third is to form multicellular organisms. Now, we can see that they've put in both fully grown, so that means that we can't use the growth as an answer. And for multicellular organisms, if it's forming multicellular organisms, it's already formed, so we can't put that as an answer too. So our answer would be for repair, but we need to be a bit more specific than that. What is being repaired? It's repair of any damaged tissue. Remember that we can't say repairing cells. What happens in mitosis is there's a replacement of cells. So cells get replaced and tissue gets repaired. Question 3. Amylase is an enzyme that digests starch. 3.1. Which organs in the human digestive system produce amylase? Tick one box. So remind ourselves about enzymes and the locations of production. So we've got to look through our options here and then just figure out which ones don't apply, and it can help us narrow down our answer. So first one, it says liver, small intestine, and large intestine. Well, the liver produces enzymes, but none of those that are involved in digestion. So it can't be the liver. The large intestine also doesn't produce any enzymes, so that definitely can't be right. But small intestine does produce some enzymes, so we should be expecting that later. So it's definitely not the first one. Second one, salivary glands. Yes, that does produce enzymes, and that actually does produce amylase. The stomach, yes, produces enzymes, but not amylase. And again, we said liver is not an option, so it can't be the second one. Let's look at our third option. The salivary glands, yes, produces amylase. Pancreas, that also produces enzymes and amylase. And small intestine, yes, that does too. So that should be our answer. Just checking the last one quickly. Stomach, no, we mentioned. Pancreas does, and large intestine doesn't. A student investigated the effect of pH and the activity of amylase. This is the method used. Prepare amylase at solution pH 5. Mix the amylase solution with starch in a boiling tube. Remove a drop of the amylase starch mixture every 30 seconds and test it for the presence of starch. record the time when all the starch has been digested. Repeat steps 1 to 4 using amylase solution prepared at pH 6, then at pH 7, and then at pH 8. So we've got a lot of information here. It's been specific in values also. So let's read on. 3.2. What was the independent variable in this investigation? 1 mark. So we've got to remind ourselves, what does the term independent variable mean in an experiment? It means what you change. So reading the information that's given to us, what is being changed? Well, they've said pH 5 we're starting with. Then later on in step 5, they said pH 6, 7, and 8. So there they've told us that we are using different pHs there. But if you just look at the first sentence, it can also give us an indication of what our independent variable is. It says a student investigate the effect of pH on the activity of amylase. So they want to see, how does this pH cause any change to the activity of amylase? The only way they could find that out is by changing the pH. So that has to be the answer. 5.3. Describe how the student would know when all the starch had been digested. 1 mark. So back to our method, it does say in step 4, record the time when all the starch had been digested. But in the previous point, it said test it for the presence of starch. So that's quite vague, and it's because they want us to recall how to test for starch. So we're thinking about our food tests right now. So again, testing for the presence of starch is specific substance that is used. And we can use our lovely mnemonic here to help us, bobbies. So what do these letters mean? The colors of our chemicals: brown to orange, blue to black. We're using iodine solution to test for starch. So if starch is present, it will turn from brown orange to blue black. But if starch had been digested, then it wouldn't be able to turn blue black. And that should be our answer. So the iodine would not turn blue black. You could then also say as an alternative that the iodine would remain brown orange. 3.4. Figure 6 shows the student's results. So we've got a line graph here with pH on the x-axis and our time taken to digest all the starch in minutes on our y-axis. You can see it's giving us our unique curve here. What was the optimum pH for the amylase? Use figure 6. So we're looking about the term optimum. Optimum means best. So we've got to figure out which is the best pH for this amylase to work. Well, the best pH would mean that it would take the shortest time for the starch to digest, so it'd be the quickest. So that means that it should be the lowest value of time on our graph. So we are looking for the point right at the bottom there that's closest to the x-axis. So reading down to the x-axis, it should be a value of 6.8. A scientist did a different investigation. This is the method used. Prepare amylase solution at the optimum pH. Mix the amylase solution with starch in a boiling tube. Measure the concentration of sugar every 10 seconds for 2 minutes. Figure 7 shows the scientist's results. So we've got a different line graph now. On our y-axis this time is concentration of sugar in arbitrary units, with the time in seconds on our x-axis. 3.5. How much time did it take for the amylase to digest all the starch? Use figure 7. So we are then thinking about if the starch is digested, then our concentration shouldn't change anymore. So we're looking for when it's horizontal, specifically when does it become horizontal. So we'd have to look very carefully on our graph now to figure that out. So when is the moment that it becomes horizontal? Looking carefully, we can see that it's going to be a time of just after 80 seconds. So you can say 82 seconds is where all the starch had been digested. 3.6. Determine the rate of sugar production per minute at 40 seconds. So for this question, what we are going to need to do, because they've told us a specific time value and because the line is curved at that point, we are going to have to use a tangent to be able to find out this rate. So what is a tangent? A tangent is a straight line that is added to any curved portion of a graph. So we find the time value that is important here, 40 seconds. And then what we want to do is you want to be able to make a line straight, which is touching the curve at that time period of 40 seconds. So we've made our tangent line now. So what that means is that at this point, we are essentially able to make and find the gradient. A gradient can only be found when there is a straight line, so we've manufactured a straight line and are now going to find our gradient. So once we've done our straight line, we are then able to make a small triangle, which is going to enable us to find 2 points on our x-axis and 2 points on our y-axis. Now remember that the formula for gradient is the change in y divided by the change in x. So we want to find the difference in two values on our y-axis and then divide that by the difference in 2 points on our x-axis. So reading of our x-axis first, our first point, our highest x value will be 65, and then our lowest will be 22. For our y-axis, our lowest y-axis value will be 2.8, and then our highest y value will be very specific, 4.35. So simplifying those now, it'll be 1.55 divided by 43, and that will be our value of rate, 0.036. However, that, if it was to give a unit, would be arbitrary units per second. But as the question asked us, we need to find the rate per minute. So what we would need to do then is we need to multiply that value by 60 to get it per minute. And then we'd come out with a value of 2.16 arbitrary units per minute. And that will be our rate value. Now remember that everyone is going to do their tangent slightly different. They're going to do their gradient slightly different. So there will be a range of answers given to you whenever you do a question like this. So as long as your values that you get are within the range, then it should be acceptable. 3.7. Explain how the structure of enzyme molecules is related to the effect of pH on the activity of amylase. 6 marks. So this can be a tricky question because we need to talk about the enzyme structure itself and how that changes depending on the pH. So let's remind ourselves of the basics of enzymes by just drawing a typical diagram of an enzyme. So what's special about this enzyme? It has a special part of it which is called an active site. And the purpose of the active site is to fit and bind to a substrate. Now, a substrate, in our case of this question here, can be our food molecule, which is starch. Now, what are enzymes made from? Enzymes are made from proteins. And the things about proteins is that they have specific shapes. So the shape of this active site is supposed to only fit starch. But there are two things that can affect how well an enzyme works. One of them is pH and one of them is temperature. Of course, we're only focusing on the pH here. So let's go to our graph now and let's think about how this relates to our enzyme. So at the optimum, which we said was 6.8, our enzyme shape should be good enough for the starch to fit and bind. But when we go to pH 8 or pH 5, where the time it takes to digest is slower, we can then figure out then there must be a problem with our enzyme. And what has happened is that the active site has actually changed shape, so it doesn't fit the starch anymore. At pH 6, for example, the enzyme active site has changed shape slightly, but it's still able to fit the starch, which is why it's not as slow as pH 5 or 8. So what we've got to do now is we've got to put all of that information into words. So we can start with our general information here. Enzymes are made from protein, and they have this specific shape. And when these enzymes are able to fold, they can make an active site, which is going to be specific to fit a certain substrate. Okay, so now we can start to talk about amylase, because of course that's what the question is asking us to talk about. So amylase is a specific active site for starch. At the optimum pH, and we can just state what it is, pH 6.8, the starch will fit into the active site and then it will get broken down quickly by the amylase. Away from this optimum, the structure of the active site will change slightly, so starch is not able to fit as well properly. So that is what causes the rate to be slower. But at pH 5 and pH 8, which is the furthest away from the optimum, the active site changes shape so much. And what we actually call this is the enzyme becoming denatured. And then this means that the enzyme cannot fit the substrate. And that means that starch won't get broken down properly, even at all. So the rate decreases and is very, very slow. So to summarize, we've spoken about what the enzymes are made from. We've spoken about how the enzyme actually works. We've then made it specific to the question about amylase and starch. We've then spoken about what happens at the optimum pH, best pH for the enzyme. And then we've spoken about what happens away from this optimum and how that affects the rate of reaction for amylase.